We're going to talk about correcting genetic mutations. Let's dive into this. Now, in my last video, we talked about epigenetic factors and how those can switch genes on or off. In this video, we're going to talk about what happens when there is a mutation to the DNA sequence itself. And just like a typo can change a word and potentially the meaning of a sentence, a mutation can change the function of a gene and ultimately affect a person's traits. But unlike typos in a book, which can often be corrected, not all genetic mutations are so easy to fix. For example, a missense mutation in the gene for hemoglobin, the protein that carries oxygen in red blood cells, is responsible for sickle cell anemia. Sickle cell anemia is a group of disorders characterized by the presence of abnormal hemoglobin in the red blood cells. An insertion is a mutation that adds one or more nucleotides to a gene. A deletion is a mutation that removes one or more nucleotides from a gene. Both insertions and deletions can cause frameshift mutations. Gene therapy is a treatment that involves introducing genetic material into a person's cells to replace or supplement a defective gene. Cystic fibrosis is a disorder that affects the lungs and digestive system. While there are still challenges that need to be overcome, the future of these therapies is bright. As our understanding of genetics continues to grow, we will develop new and innovative ways to treat genetic disorders. Who knows what the future may hold? Maybe one day soon, we will be able to cure diseases like cystic fibrosis and sickle cell anemia. If you have any thoughts on this, please share them below. As always, the references and studies used are listed below. People with cystic fibrosis have a mutation in a gene called CFTR. This gene provides instructions for making a protein that regulates the movement of salt and water in and out of cells. People with cystic fibrosis have a mutation in the CFTR gene that causes them to produce a protein that does not work properly. This leads to a buildup of thick, sticky mucus in the lungs and digestive system. There are different types of mutations that can cause cystic fibrosis. The most common type of mutation is a deletion of 3 nucleotides that causes the CFTR protein to be non-functional. However, there are other types of mutations that cause milder forms of the disease. One approach to treating cystic fibrosis is to use gene therapy to replace the mutated CFTR gene with a healthy copy. Another approach is to use gene editing to correct the mutation in the CFTR gene. Both of these approaches are still in the research stages, but they show promise for treating cystic fibrosis. Gene therapy is used to treat a variety of genetic disorders. One example of gene therapy is the treatment of severe combined immunodeficiency (SID). SCID is a disorder that affects the immune system. People with SCID have a hard time fighting off infections. In the past, the only treatment for SCID was a bone marrow transplant. However, in the 1990s, scientists developed a gene therapy trial for children with a form of SCID called X-linked SCID. X-linked SCID is caused by a mutation in a gene on the X chromosome. This gene is important for the development of T cells and natural killer cells, which are both part of the immune system. In the gene therapy trial, children with X-linked SCID were given healthy copies of the gene via a retrovirus vector. The virus was modified so that it could not cause disease. The results of the trial were very promising. The children who received the gene therapy had a much better chance of surviving and living a normal life. A frameshift mutation occurs when the reading frame of the gene is shifted, which changes the order of the codons and the amino acids that they code for. Frameshift mutations can be more harmful than other types of mutations because they can completely change the amino acid sequence of a protein. This abnormal hemoglobin causes the red blood cells to become sickle-shaped, which can lead to anemia and other symptoms. The first approach that I want to discuss is called gene editing. Gene editing is a new technology that allows scientists to precisely alter DNA sequences. There are many different types of missense mutations that can cause sickle cell anemia. However, all of them result in the replacement of glutamic acid with valine at position 6 of the hemoglobin beta chain. This single amino acid change alters the shape of the hemoglobin molecule and causes it to form long, rigid fibers. These fibers distort the red blood cells and make them feel like sickles. People who have 2 copies of the sickle cell allele are said to have sickle cell anemia. However, Those who have one copy of the sickle cell allele and one normal allele are said to have sickle cell trait. Individuals with sickle cell trait typically do not experience any symptoms of the disease unless they are exposed to low oxygen levels. However, carriers of the sickle cell trait are resistant to malaria, which is a serious and sometimes fatal disease caused by a parasite called Plasmodium falciparum. The parasite is transmitted through the bites of infected mosquitoes and affects red blood cells. It is thought that the sickle cell trait confers protection against malaria because the parasite cannot multiply effectively inside sickle-shaped cells. This is thought to be one reason why sickle cell anemia is more prevalent in areas where malaria is common. However, scientists have been working hard to develop new methods for correcting genetic mutations, which could one day lead to cures for currently incurable diseases such as cystic fibrosis or sickle cell anemia. This results in a protein that is shorter than it should be. Nonsense mutations can be more harmful than missense mutations because they can lead to the production of non-functional proteins. Some nonsense mutations, however, do not result in severe disorders. So today, we're going to discuss some of the most promising approaches to correcting genetic mutations and explore the ethical issues surrounding them. Now, what is a mutation? Well, a mutation is simply a change in the DNA sequence. This can happen spontaneously at any time, or it can be caused by an environmental factor like a chemical exposure or radiation. Regardless of how they occur, these mutations are the ultimate source of all genetic variation and disease. Mutations can range from small changes, such as a single nucleotide-based substitution, insertion, or deletion, to large-scale changes, such as inversions, translocations, or whole chromosome aneuploidy. Now, if you think about a person's genome as their unique instruction manual for building a human being, then a mutation would be like a typo in that manual. Alright, let's go back and review some basic genetics. A gene is a segment of DNA that contains the instructions for making a protein. Proteins are the workhorses of the body and carry out a wide variety of functions. The sequence of DNA bases along a gene determines the order of amino acids in a protein, which in turn determines the protein's three-dimensional shape and function. Sometimes changes occur within the DNA sequence and alter the genetic code. These changes are called mutations. As I mentioned before, mutations can be small or large, but they can also be beneficial, neutral, or harmful. A neutral mutation is one that does not change the amino acid sequence of a protein because of redundancy in the genetic code. There are 64 possible combinations of the 4 nucleotide bases, read in groups of 3, but only 20 different amino acids commonly found in proteins. This means that multiple codons often correspond to the same amino acid. For example, both the codons GCU and GCC code for the amino acid alanine. Therefore, a mutation that changes GCU to GCC would not change the amino acid sequence of a protein and would be considered a silent mutation. A mutation that changes a codon to one that codes for a different amino acid is called a missense mutation. Another type of mutation is a nonsense mutation. A nonsense mutation is a mutation that changes a codon to one that codes for a stop codon. The final type of mutation that I want to mention is called a frameshift mutation. Insertions and deletions are 2 types of frameshift mutations. So now that we have reviewed some of the basics of genetics, let's take a look at some of the approaches that are being taken to correct genetic mutations. The next approach that I want to discuss is called gene therapy. Another example of gene therapy is the treatment of cystic fibrosis. Gene therapy and gene editing are 2 powerful tools that have the potential to revolutionize the treatment of genetic disorders.